Okay, everyone, so we're going to get started. My name is Mark Lamb. I'm a product manager here at Creme Global, and with me is Cian O'Mahon, who's our Chief Science Officer, and you'll hear from him in just a couple of minutes. So just a few things about the format of today before we get started. So Cian is going to do a presentation, and he's going to step you through it, and then at the end, I'm going to do a more practical demo in the software. So we're going to start that, but just a few things. There is, if you have any questions, there is a chat window you can open. So if you just move your mouse to the top of the screen, there's a few green buttons. One of them is a chat window. Anything you type in there, that's how we'll be taking questions. Everyone else's mic is muted, so we won't be able to hear you. And also, anything you post in the chat window is public, so anyone in the webinar will be able to read it. But if you've got any questions, just throw them in there, and we'll look at them as we go through. Like I said, we are recording this, and we will try to make it available as soon as we can afterwards, so that you can catch up on it again. So with that, I'm going to hand over to Cian, who's going to do a presentation.
Thank you, Mark. So good morning and good afternoon to everybody joining us from different parts of the globe. So I'm just going to start with a very quick overview of Creme Global as a company. We have been in operation since 1999, actually, was the start of the company, which was when we spun out of a research project in Trinity College Dublin in the Centre for High Performance Computing there, so the TCHPC, the Trinity Centre for High Performance Computing. The company was formed in 2005, and since then we've gone on to develop different products and services for the food, chemicals and cosmetics industries in a number of different areas. So back in 2000, I don't know, that's not coming up clearly on the slides, but back in 2008, we launched a number of different products. We've gone on to develop a product called Creme Food Safety. We have a product called Creme Nutrition. We've also products in the area of Creme Care and Cosmetics, looking at consumer exposure to ingredients and personal care products and cosmetics. We develop a number of different products in the area of personalized nutrition, as well as general consumer exposure and food safety. And we deliver all of this through a data science platform called Expert Models that Mark is going to tell you a little bit more about at the end of my presentation. So just an overview of what I'm going to talk about. So I'm a mathematician by training, but I've worked with the food, chemicals and cosmetics industries my entire career. So I'm going to talk about food safety and how data science has found a place within the broader question of how we make sure that we have safe products for consumers in the marketplace. So I'm going to talk about what it means, the differences between data science and science broadly speaking. I'm going to talk about predictive microbiology. I'm going to talk about genomics and some of the more interesting developments then around the Internet of Things, blockchain technology, and a bit on chemical food safety and consumer exposure assessment. Okay, so jumping in, I want to start with a kind of a philosophical question. That's something that's been on my mind for quite a bit, which is, what is the difference between science and statistics and data science? Or what does it mean when we apply data science in science broadly? Because, you know, I'm a mathematician by training. I work primarily with scientists, but I work for a data science company. So what is the difference between all of these different things? So when we think about science, we think about the scientific method. And when we think about it as a process, we usually start with some observations about some sort of phenomenon in the broader world that we want to think about. We ask some questions about it. We formulate some hypotheses that try and explain what we are seeing. We develop testable predictions, gather data to substantiate those predictions, and refine that process until we can develop a general theory which explains something about the world around us. So that's the scientific method when we think about it as a proof. Then thinking about data science. Well, this is from the Harvard online course in data science, and this is saying, okay, you ask an interesting question, you get some data to try and help you answer that question, you explore the data and see what you can learn, you model it, so you develop some sort of algorithm or some sort of predictive model that explains the data, and then you communicate and visualize the results. So a data scientist is somebody who has a skill set in one or more of these little boxes in a flow. And this is you know, much broader than applications in science. This could be, wondering about, can I better respond to different queries based on questions customers are asking in a call center? Or can I, optimize how I reach different customers in a marketing campaign by measuring responses on social media? So I was hitting my head against a wall trying to figure out what the difference between the two of these is, and in the end I decided that it doesn't matter. Both of these have merits and strengths, and they're entirely complementary. And the key point that I want to drive home is that in the data age, our ability to gather, capture, store, and analyze data like never before has expanded the opportunities for science. And in particular, I'm going to talk about some of those opportunities for food safety. And that's the key point. We're swimming in data now, and the question is, how can we leverage it in a scientifically rigorous way to answer questions about food safety? But before I do that, I want to talk about how this can go a little bit wrong. And I'm going to talk about an interesting question, which has been dubbed the parable of Google flu. So a couple of years ago, Google launched a commercial product called Google Flu Trends, and the idea behind this product was that when people get influenza-like symptoms, they start Googling those symptoms. And if we can link the rate of how people are searching for those flu-like symptoms to the rate of influenza that's being measured in the population, we can use search terms to predict what the incidence of influenza is like in a particular population of consumers. Now, Google went ahead and did this, and they developed a model by taking 50 million search queries, linked them against about 1,000 data points, and they correlated it, or they linked it to the actual measured influenza rate that was reported by the Center for Disease Control in the US based on their lab data using CDC data. And they used a logistic regression type model using the kind of schematic equation you can see in the bottom corner. Now, a guy called David Laser published a paper back in 2014 analyzing how well Google Flu Trends was actually predicting. And what he found was, in certain cases, it was predicting more than double the CDC estimates. So why was this happening? And furthermore, he found that if you used a simpler model that just looked at 2 weeks of lagged CDC data, you could predict what was going to happen in the future with a lot better accuracy. So even though Google were sitting on all of this incredible data that in principle should be able to tell us a lot about the rate of influenza in the population, the algorithm itself wasn't behaving that well. So why was this happening? Well, one of the first things that we learned from this is that the Google search engine is originally developed for any kind of scientific purposes. And we take a step, all empirical research rests on a foundation of good measurement. But the Google search engine is always changing. They're always changing the algorithm and changing how they measure search terms in the population. So that means that the measurement is changing. So they're leveraging from a moving target, and that confounds your algorithm. So without going into all the technical details, some simple learnings is that we can use big data to understand the unknown. But if we have good data that's smaller and explains things well, that's perfectly adequate. Like in the example of Google Flu Trends, we found that the CDC data actually predicted 2 weeks out what the influenza rate was better than any of the search terms. You need to know the algorithm in a lot of detail. So how well is it predicting? What are the assumptions it's making about your underlying data? The size of the data is not the only thing that counts. Just because you have big data doesn't make you super powerful, especially when you're trying to answer scientific questions. And it also raised a lot of questions around replicability and transparency. So any scientific hypothesis or theory needs to be repeatable. Google didn't publish what the actual search terms they used to predict influenza was. So that meant that people couldn't duplicate their findings. And that, so how well it's behaving, you have to have a lot of faith and trust. And that's difficult when, everything is not being really being revealed in a scientific publication. But on to more successful cases and more applicable examples in food safety. So the first one I want to talk about is predictive microbiology. So this is an example of a predictive model we developed for a company called Hawkins. Hawkins make antimicrobial ingredients that, for example, this is one that they make called Eliminate, which is used to control the growth of Listeria monocytogenes in ready-to-eat foods. So it's a predictive model. It takes a number of fairly simple inputs: the moisture content of a ready-to-eat meat, the salt content, the pH, and the concentration of their Eliminate antimicrobial. You put in your initial concentration of Listeria, the prediction time, the temperature that you're going to predict at. So to develop these type of predictive models in microbiology, we divide them into primary models, which are mathematical models that describe the growth of bacteria as a function of different mathematical parameters. Secondary models then link those mathematical parameters to your environmental conditions, so things like time, temperature, salt content, pH, water activity, and so on. And then tertiary models are like your software implementation of a predictive model. So when we run this particular predictive model, we see that you get two curves back. The black curve is what happens if you don't have any antimicrobial ingredients. The green curve is what happens if you use the Eliminate product. So this is a very nice way of predicting bacterial behavior. It's based on scientifically published models, and it can be used to proactively formulate safe products and inform how you should formulate a product with a given antimicrobial. What has revolutionized the area of microbiology in the last number of years is genomic data. And this is a very famous graph at this point, which shows how the cost of sequencing an entire genome has vastly, in the last number of years, outpaced Moore's law. It was tracking quite closely for a number of years, but you can see it's vastly outpaced it. And the cost is now even less than $1,000. It's down to a couple of $100 to sequence an entire genome. So traditionally, when we're developing predictive models, we group bacteria together, we quantify growth and survival in some numerical method, and we consider the behavior of different bacteria separately, and we don't use genetic information in any real way. With omics, now we can fine-tune these predictive models, and I'll present some examples of how we can do that. We can understand food ecosystems and their dynamics, and we can quantify strain variability. So we know that within a given species, different strains can behave in very different ways. And omic data, like whole genome sequencing, allows us to differentiate. So a couple of compelling examples of why this is important. So this is work published back in 2008, looking at Bacillus cereus and how it's been divided into 7 major phylogenetic groups. So you can see there down at looking at group 6 at 7 degrees. You can see at the bottom left-hand corner, 100 strains in group 6 will grow at 7 degrees, while in groups 7, 3, 4, 1, and 5, no growth is registered. Conversely, at the other end of the scale, at 50 degrees, in group 7, 100% of strains in that group will grow at 50 degrees, however, the rest of them don't. So you can see that the amount of variability, if we grouped all of these together in a predictive model, we would, the uncertainty or the range of responses we would be seeing is absolutely huge. So genetic information allows us to subdivide and be a lot more targeted in how we develop predictive models. Another example is looking at specific biomarkers that can tell us how different bacteria are likely to behave. So this is dividing B. subtilis into different groups, and you can see that there's a genetic element, and we can identify the specific genetic element and see how it affects thermal resistance in some groups but not in others. So you can see marked differences in the Z values. And again, knowing this differentiation can have a massive impact on how you design your food production processes, and bringing this knowledge into a predictive model then tells you how you can design safe products. So that's looking at whole genome sequencing to subdivide our bacterial strains into different groups and understand how they behave. Another massive use of omic technology is the use of metagenomics, or looking at the broader microbiome. So this is an example from 2014, where we have published by Ed Fox et al. If people are interested, by the way, I can send on references to all of these different publications afterwards. So they looked at a meat production facility, and they looked at different Listeria species that were present in different drains across the facility. So they examined 4 drains in the facility. They found that 2 were Listeria positive and 2 were Listeria negative. And they wanted to explain this using the microbiome of the different drains. So using metagenomic data to look at the relative abundance of different bacterial species and so on. So the microbiome analysis really, this is at the class level and the family level. On the left-hand side, we have the negative Listeria negative drains, and the right-hand side we have the Listeria positive. So you can see that broadly speaking, we have the same classes, the same families, and there's no major qualitative difference between the relative abundances of different classes and families. But going a bit deeper, I just want to draw your attention to the top right-hand corner of this graph. So the black square are the Listeria positive drains and their overall diversity. The black circle are the Listeria negative drains and their overall diversity, measured in operational taxonomic units. And you can see that the overall diversity of the Listeria positive drains is much, much higher. So that suggests that there's something going on here. So they investigated this in a lot more detail. And looking at the microbiome, they found that there are 21 families that were present only in the Listeria positive drains. And 3 families in particular were all associated with a higher abundance in the Listeria negative drains. So they wanted to confirm that there was a protagonist-antagonist effect here. So they did some co-culturing experiments, and they found, yes, Xanthinobacterium lividum reduced the attachment and biofilm growth of Listeria monocytogenes, while E. gallinarum caused an increase in biofilm formation of Listeria monocytogenes. So what's this telling us? This is telling us that the influence of the broader environment and all the other bacterial species that live in different parts of the production facility actually have a massive effect on the prevalence of pathogens that we're trying to control. So again, and it suggests a control strategy that we can use. So if we can identify drains that are colonized by Listeria and drains that are free, we can and profile the microbiome. And this, you can use different technologies like shotgun metagenomics or 16S metagenomics. You can determine what the potential protagonists and antagonists are. You can look at, you can isolate them, determine the effect they have on Listeria attachment and biofilm formation. That's using your co-culturing experiments. And you can pathogen in-house, that's crucial, so it's local to the different microenvironments you have in the production facility, and use them to control the prevalence of Listeria colonization. And this means you can do it without the use of, or at least a reduced use of antimicrobials in the production facility. So this brings us towards an idea of ecosystem modeling. So whereas before we had a predictive model that told you how a pathogen grew in a given environment, and you had a safety objective that you wanted to meet, you predict whether under different conditions your pathogen grows or is thermally inactivated, for example, and whether you're above or below your food safety objective. And you just look at one bacteria at a time. Now we can look at the broader ecosystem that the bacteria lives in, and we can look at different scenarios. So like this is a schematic, but you can see in the top of the slide you have bacteria A and B and a pathogen P, and you can see in the presence of A and B the pathogen grows and our food safety objective is not met. So that is not a desirable situation. In the bottom of the slide you can see that you've got innocuous bacteria A and C and the pathogen. These have an inhibitory effect on the pathogen and our food safety objective is met. So the potential for this type of analysis and technology is massive, and its implication for data science and predictive modeling is also huge. So one project where we're taking this challenge head on is called the SAFE project. It stands for the Sequencing Alliance for Food Environments. And it's a 3-year project that's being led by University College Dublin in collaboration with ourselves, Creme Global, with 6 industry partners, Kerry, Glanbia, Dairygold, Dawn Farms, Mead Johnson, and Nutrition Supplies. And it's funded by Enterprise Ireland and coordinated by Food for Health Ireland. So we have 5 production facilities that we're studying over 3 years in the project, and we're developing ultimately dashboards that companies can quickly use to understand what's going on in their food production facilities at a level of detail that's never been seen before. So, for example, they can have a dashboard of samples, and from using molecular surveillance within the facility, they can see what time of year the samples were taken, the sample number, and they can look at what, using whole genome sequencing, like you can have a traffic light system where you have different traits that can be identified for the different isolates coming from the sampling plans. You can see, have we got antimicrobial resistant bacteria? Are they particularly virulent? Are they showing traits of thermal resistance? Are they likely to form biofilms, and so on? You can easily look at the microbiome of the environments that these bacteria live in, where they come from, and again, what are the likely effects these broader environments are having on the prevalence of the different bacteria in the facility. Okay, so that's just a whirlwind tour of the potential for genomic data in food safety. I think, depending on your area, you may be familiar with this, but it's definitely presenting some really exciting challenges and opportunities for data science and predictive modeling at the moment. Another really interesting area that's generating a lot of data is the Internet of Things. So the idea that any physical device can be connected to the internet or connected to some sort of server and can be transmitting and communicating information in real time. So an example is sensor data in food production. So you can be doing environmental monitoring. You can be in real time monitoring the time, temperature, pH of your food production environment. You can be monitoring the actual food matrix itself, depending on what you're producing. One of the really hot areas for IoT at the moment, and the data that goes with it, is the idea of predictive maintenance. So can you predict when your equipment is likely to fail, and can you do that using the data that's generating in real time? Also, of course, the question is, can we use this data to predict, or can we use it in risk assessment modeling or analysis? Based on food production, or even can we do it based on the supply chain, as things are transported from the farm to fork supply chain continuum. There's also lots of really cool innovations in the area of precision agriculture. So the idea of monitoring animal health in agriculture, looking at how we can monitor, even just time, temperature in real time for different animals can really help us optimize their things like their body weights, their milk production in dairy, and so on. If you're interested in tillage, you can monitor the health of soil in real time and look at all things like water activity. You can look at environmental monitoring, can look at like the climate at a given point in time and make some change. A bit of a fun example of how this that we've implemented in-house in Creme Global, and just downstairs from us is a cool company that offers IoT solutions called Denalto. You can check out their website afterwards if you're interested. But they gave us one of their IoT sensors, and we've installed it in our fridge. So this is the Creme Global fridge, and you can monitor temperature and humidity in real time using one of their dashboards. And just last Friday, we had a party in the office, as we often do at the end of the day. So I can zoom into this data and I can look at massive deviations in temperature of the fridge. So you can see that, it's normally at around 3 degrees, but I can see oscillations going up at 9 degrees and then up at 15 degrees. So I was wondering what was going on. Well, it turns out in the morning, some of my colleagues on the social committee were loading beer into the fridge, and as a result, we're opening and closing it and adding a lot of mass into the fridge, which raised the temperature of the fridge significantly. Over time, the temperature went down, but then when the party actually started, people were going to and from the fridge back and forth the whole time, and as a result, the internal temperature of the fridge went as high as 15 degrees. So this doesn't pose massive food safety issues to us because we were only drinking beer, but you can see how having this real-time information allows you to understand what the cause and effect is of different food safety considerations in a given, food storage facility or food production. The real opportunity here is to come up with smart systems. So not just use the data for monitoring, even though that is really, really helpful, and not even just to develop predictive models, and those are really important as well, but you can develop the models in such a way that they suggest what you should do in light of what you're monitoring. So, that they're suggesting corrective action that you can take in real time. So, if we had information saying you really shouldn't open that fridge for at least an hour until the temperature can be reduced enough, or maybe that it would correct and say, we need to reduce the temperature by several degrees now in order to compensate for how it's going up. That's the real opportunity, in my opinion, for these kind of sensor IoT data and how it feeds into decision making and actually suggesting corrective actions. That's the real value add. So again, moving on to another really trendy technology at the moment, which is called blockchain. People might have heard of it. Originally it currently is the technology that underpins the Bitcoin currency. The whole idea is that it's an open ledger of distributed transactions. So it's a digital list of records, which are called blocks, that are linked using a specific cryptographic algorithm that I won't go into the details of. But the whole idea is that it's extremely secure, it's open and distributed, and it's very, very difficult to modify or change. So the opportunity that presents is for food traceability and food safety. So IBM have a massive partnership with lots of major food production facilities, as well food companies, as well as Walmart. So people might be familiar with a guy called Frank Yanis, who's a big proponent of this technology, and they're using it to improve the traceability in their supply chains. So one of the massive challenges for any food company or food supplier is in the event of an outbreak of foodborne illness, is doing your track and trace back. And blockchain allows us to very, very quickly trace back our food chain, our supply chain, and understand, what the source of a particular outbreak is, because we use the blockchain technology to very transparently and rigorously track your supply chains, and also, check what are the conditions in your supply chain, what are your time, temperature, storage conditions, what's the humidity, and so on, what's the integrity. So this is one of the things that people think at the moment are poised to revolutionize how we do food traceability. So this article that you can see, this is a news release that goes back only to August on IBM's website. So all I'll say is watch this space. This is really poised to do some exciting things. Okay, so I'm going to shift gears again a little bit, and the analysis of food consumption data. And the question that I'm posing very, very simply is what do people eat? And if you think about it for a second, the question of what do people eat is a very complicated one, because people consume different foods in different amounts, in different combinations, in different geographies, at different life stages all around the world. And accurately capturing this information is a very, very challenging thing to do. So, and the benefit of being able to accurately capture people's diets has many, many different applications. For example, you've got applications in chemical food safety, making sure that all the different additives, flavorings, contaminants from food packaging, environmental contaminants, pesticides, and so on are safe at the levels that they exist in different foods. It's very, very valuable in food epidemiology if you're looking at the incidence of foodborne illness in a population, and has huge applications in nutrition that I won't even go into today, but that could be something for another webinar. So going into the question of food chemical exposure, then looking at how people are exposed to a given chemical is very simplistically put here on the slide. You need to know the amount of food consumed and the level of chemical in the food consumed to look at the dietary exposure. You usually do this to generate a distribution of exposure in the population. So just like I was saying, everybody consumes different foods in different amounts, so everybody has a different level of exposure to a given chemical. So the exposure in a population is some sort of distribution. You usually measure that exposure in milligrams per kilo body weight per day, and that's because we compare it to a safe level or a reference dose, which usually comes from toxicology, and see what the risk is in the population as a result of the chemical exposure. And this is a question that has implications reaching far beyond food. So, a given chemical like a flavoring could be present as a fragrance in personal care products and cosmetics or in air care products. An additive could be present in our medication. A pesticide could be also something we're exposed to as a result of using different pesticidal products in our gardening. We can have the same environmental contaminant and exposed from like the air we breathe, in dust, in soil, in food. So understanding what we call total aggregate exposure is a very, very complicated one. And a lot of what we do at Creme Global is developing refined methods to understand this question to make sure consumers are safe. And what we don't want to do is, take a worst case across all of these different scenarios or take an average across all of these different scenarios and add them up, because that gives you a very unrealistic picture of exposure. We're all about trying to come up with the most refined answer that you possibly can, and doing that allows you to make a very, very informed decision. So the starting point for this type of analysis is using food consumption data. So these are nationally representative surveys of food intake in a population of consumers. You survey the diet of 1,000 or more consumers for 1 or more days. It's usually hundreds of thousands of lines of consumption data, and usually have information about their demographic, their age, their gender, their body weights, their things like socioeconomic status, and you might even have biomonitoring data looking at level or biomarkers of different nutrients. One of the main things that these surveys are done for is for nutrition, but they get reused again and again to answer all different questions for chemical food safety. And again, it's not even called the digital age anymore, it's called the data ages, which is where we're in now. We have all of these different sources of information that we can use to carry out these types of analyses in a lot more detail. So, for example, there's a company called Kantar World Panel, there's Nielsen, Euromonitor International, NPD Group. All of these companies generate loads of information and data on things like market shares of different companies. They have their own panels of people and they survey what they buy and what they eat. And because they use digital tools to gather information, they can have far-reaching sample sizes than we ever could using manual methods to gather this information. We've also got on the right-hand side there, we've got CodeCheck and Mintel. These are like labeling databases, so we can look at knowing what's on the label of a product can tell you an awful lot about what's inside it, like what food additives are there, or what the nutritional composition is. On the left-hand side are two projects that we were involved in. One is called Foodbook24, which is an online approach to gathering dietary data, or the Food for Me project, which is a pan-European project where we're looking at approaches to personalized nutrition and even generating advice on a personalized individual level on how people can improve their dietary habits. And then down the bottom, we've got these kind of voluntary initiatives where people submit data themselves on, like the quantified self for data donors, where people just voluntarily submit data on what their habits are, what their health status is, and so on. So integrating all of these data sets presents massive opportunities for refining how we do consumer health analysis. And one of the ways that we do this is we can do probabilistic exposure assessment. So this is a way of analyzing all of this dietary data together in an algorithm to produce a distribution of exposure to a given chemical in a population of consumers. So I'm going to explain how that looks visually. So we have a big data set, and I'm going to explain one of the algorithms that we have implemented in our software, and I'm going to hand it back over to Mark, who's going to give you an actual live demo of what that algorithm looks like from a user's perspective. So The way the algorithm works is we start with a diary of food consumption information. So we go into person 1 on day 1, and we look at what foods they ate and in what amounts. And then for a given chemical, it could be a food additive, it could be an environmental contaminant, it could be a pesticide. We go to a distribution of concentration, and we take a random sample from that distribution. And that's all Monte Carlo simulation is, or probabilistic modeling. It just means doing random simulations of statistical distributions on computers. And we can calculate that person's total aggregate exposure to the dietary chemical on that day. We repeat that for however many days of food consumption that person has registered. In this example, I have 2 days per person in the database. And we do that for every other person in the population. And this 2 days may seem like very little, and you could argue that it is, but it's the 2 times 24-hour recall method is actually a very standardized method of gathering dietary information. The National Health and Nutrition Examination Survey in Haines in the US, for example, has 2 days of food consumption per person. And then we can calculate their daily average exposure to the chemical for each individual. And that's usually what we compare to a chronic reference dose from toxicology. We can also look at their acute exposure, so that's each single day of exposure, which is like the peak of exposure, and you compare that to something like an acute reference dose. And you do that thousands of, tens of thousands of millions of times. These are called iterations of a probabilistic model. You do that for everybody, and you generate a virtual population of consumers that's all leveraged from real data, and you rank them from lowest to highest. And that's all the distribution is when you think about it. It's a load of numbers from smallest to biggest. And you calculate statistics on that distribution. For example, you can look at the mean exposure in the population, you can look at the median, or you can look at the P95, which is one of the common standards for risk assessment that we use in the European Union, for example. So that's what it looks like to generate this type of information. And one of the things we've been doing an awful lot of work in at Creme Global in the last number of years is developing production versions of these data science type models that are very, very easy for a non-data scientist to use, or even for a data scientist who doesn't want to be running code all the time but wants to use a front end to be able to run their analyses quickly and easily. So that gives me a nice segue into our Creme Food Safety software, which is on our Expert Models data science platform, and Mark is going to tell you a little bit more about it. But maybe before I hand it over to Mark, actually, are there any questions people would like to ask on anything I've presented so far? And everybody is on mute, so can I request that you use the chat if you want to ask any questions? So I'll leave it a couple of minutes in case people are typing. And Mark, do you want to start talking, get set up in case?
Yeah, well, I'll jump into it maybe, and we'll pop back open the chat window in a couple of minutes and see if any questions have popped up. So, okay, like Cian said, I'm going to do a demo of the software. So I'm just going to jump into my browser here. Okay, so I will start here. Okay, so I'm just going to give a little bit of background of what kind of software this is, and then I'm going to jump into a demo that's similar to what Cian was describing in the last few minutes of his presentation. So models online, so you can access them through the browser. Typically, one of the most important use cases for it is what Cian was talking about a few minutes ago, that non-data scientists need to be able to run a model, and they need an interface into a model without running it on the command line or running it in the way that a developer or a technical data scientist would in a Python environment or whatever it is. So Expert Models allows the models to be hosted in the cloud, so people can easily access them through the browser. And it also exposes the parameters that people might need to put into the model. So they need to input different types of data, which we're going to look at in a few minutes. They need to change some parameters like the number of iterations they want or different types of calculations that they want the model to do. And it makes this very user-friendly instead of having to specify these, again, through some technical environment that a technical data scientist or a software developer might do. Now, I think we do have one question on the. So we have one question on the slides. Will they be available afterwards? So we are going to make a video of the webinar available afterwards, and obviously the video will have Cian going through the slides. Okay, so thank you for that question. I will check the questions again every few minutes, I guess, and at the end of the demo, I will go through some answers. So if you do have any questions, just pop open that chat window at the top. It's just the kind of green icon at the top of your screen, and it's got a chat bubble, and you can type them in there. Okay, so with that, I think I'll start the demo. So this isn't too long of a demo. It's fairly shallow, I guess. This is obviously a pretty complex area. The demos can be as long as we want them to be, but this is a pretty quick run through really what Cian was describing in the last kind of 5 minutes or so of his talk, looking at that kind of exposure assessment, because that's one of the models that we have available on Expert Models. So first, this that you can see here is Expert Models. So as I said, it's a cloud-based piece of software. So I just logged in my browser, and now I'm going to firstly I'm going to open some of the data just to make sure we kind of know what we're looking at. So I'm going to just briefly open one of the dietary surveys that Cian was describing, and then I'm going to go through using that dietary survey to run an assessment. So one of the things we have in Expert Models is a large collection in this food data science folder of data sets in the area of food safety. So you can see the different countries here, and the one I'm going to use for my model, which I'm going to run in a few minutes, is this one. So I'll open it. There's a few different tables here. Many of you may well be familiar with this kind of structure already, but I also want to show how the system handles them and how they appear in the system. So I'm just going to very briefly open the main ones and maybe show a couple of things. So if I open 3 of these tables in a new tab, we just open these here. So the 3 that are very much linked together here. The first one is our subject table for this survey. So like I said, a lot of you will be familiar with it already, but there's just 1 row per subject here. So you can see each subject has a code, and we've got a lot of the information on them that Cian described a few minutes ago. The sex of the person, the age, the height, and lots more information as well as we go. And there's about 4,100 subjects in this survey. The second table is the food table. So this is the list of foods that are in the survey. So you can see it's a fairly simple categorization. We've got a name for each food, a code associated with it, and then foods are broken into groups, and these groups themselves have names and codes where there's only 1 food in each group. Finally, we've got the consumption diary. So this has 1 row per consumption event. So you can see the subject code. So it's referencing the person in the subject table I had opened a moment ago, and it's referencing the food in the food table I had opened a moment ago, and it's saying this subject ate this food, and importantly, it has the amount, which in this case is specified here. So the diary obviously gets quite big. This one is 384,000 entries, and it's got every consumption event in the survey. And these are the 3 main tables, as well as a 4th one that I'm going to show in just a minute, that the model will be using in the way Cian described as it's stepping through them. So one thing we can do if I open up so this is our subject table again. So like I said, there's 1 entry here per subject. And if we wanted to visualize this data, I suppose, just to make it a little bit more interesting, we could do something perhaps a little bit contrived, but maybe we just filter it down to only look at people who are over 18 years of age. So we're filtering to where the age is greater than or equal to 18, and then maybe very quickly we could just do something simple with a chart. So the software makes it easy enough to just visualize something. So I might just look at a histogram of the body weight in the population, something simple like that. Or maybe we could look at a bar chart, and we could group by sex and look at body weight. So you're looking at the mean body weight of people who are male versus people who are female in the survey itself. Or maybe if we did a box plot for that, it's also another great way to kind of visualize the data. So just to make it a bit more visual, I suppose, just to show how the data looks and how it's represented in this table. So now that we've seen the data, I suppose we want to jump into the most important thing, which is looking at running an assessment itself. So I have prepared some data that I'm going to use for this demo. So on top of what I've just shown you, I've created a workspace in Expert Models. So it's a handy piece of software for creating projects, because this is a common workflow amongst our users, creating folders and creating sections for projects that will be worked on over a number of months. So 2 tables I've created here. I'm not going to use the food table that I showed from the survey, because I've actually decided to only use a subsection of the foods. So I've saved this as my own table, just 56 foods I'm looking at, and I've recategorized them. So this is something that people often want to do. I've recategorized them into different groups because I just have different intentions when I'm running my assessment, as opposed to the master foods table that comes with the survey. So these are the same food codes that are in the survey and the same food names, but I've just put them into my own groups because that's how I want them to be visualized in the output. And alongside that, and finally, we have our chemicals table. So our chemicals table contains all the same food codes because we're linking this chemical, which is aspartame, to the food. So I've got one entry for each food here. I've got our chemical code, and I've got a concentration value as well here in this field for the concentration of this chemical in this food. And this is what our model is going to be run on today. So there's only 4 food groups. We're not looking at the entire diet. We're just looking at a subsection of it for simplicity's sake. Okay, so I'm going to run the model on that data. Just to describe, I suppose, one of the most important parts of doing this is the interface to the model itself, which can become quite complex. There's often a lot of parameters you need to provide when you're running the model. You might need to provide different data every time. There's lots of inputs. You might need to look for different statistics at different times. You might want the model to do additional calculations. So within expert models, when I open our chemical exposure model, we have an interface into each model. So this is a wizard that exists within the browser where people can specify exactly what they want their model to do. And this is where you set up your assessment before you run it. So there's lots of things you will tend to need to do. In this case, for this model, I need to provide 4 tables of data. They're the ones I just looked at. And then there's 2 more pages, which I'll go on to in a second. So I'll just choose my data. So my food table, I saved in this folder I was showing a second ago. And my chemicals table is in the same folder. And then my subject table is in the master data set itself. So I'm just going to open that. This is the one we were looking at a few minutes ago. Okay. Now, at this point, so I mentioned I had created another food table because I just wanted to look at a subsection, and that's something I can do quite easily. I can manipulate the data like you would in Excel, select some of it, copy and paste it out to a different table. But you can also, I can filter the data at this point. So if it was just something I wanted to do as a once-off, I could create a filter, for example, on our subject table and just look at people who are over the age of 18 or something. I'm not going to do that in this case, but you can apply filters at this point, and then you can really customize what's going to be shown in your data. So now that I've selected the input data that I want to use for my assessment, I'm going to move to the next step. So the things you can choose when you're running an assessment obviously get pretty complex, and you can do training with this model for a couple of days. It obviously gets pretty complex, so I'm not going to go into the details of everything in this demo. There's some things I'm going to just have to, by necessity, mention in a kind of a shallow enough way. But what we're doing in this section is just structuring how we want our output to look. So by default, the model will output the exposure to the subjects from all 4 of the food groups I've provided. But additionally, I can look at those individually as well. So if I wanted to see the exposure to any of these groups individually as well as to all 4 of them, I could do that. And I am in this case, so I want to see all 4 of those groups in my output. I can also stratify if I want to. So instead of just running the population as one population, I can stratify it into populations based on age groups, body weight groups, or based on sex. This is essentially like running 2 assessments in one, or with age groups, you could run 6 or 7 age groups, and it allows for easy comparison between different things. I'm just going to do that in a very kind of simple way here with a few age groups, and we'll see that in the output just to see how it looks. So I'm going to break our population into 3 age groups, which is just fairly shallow kind of look at how stratification can be used. But we've got 3 age groups here, so we'll see in the output that our output is actually, instead of just being for the total population, it's broken up into these 3 age groups, and we're going to see the output for all of our food groups individually as well. So I move on to the final stage of our wizard. There's a lot here, and there's a lot we can do. There's additional calculations that go with this model that I am going to go into in a huge amount of detail. I am going to generate this subject data that's essentially just the raw data behind the model. So as well as getting the summary statistics, which I'll show you in a few minutes, we always get the mean, the P95, etc. We also get the data at a subject level, so you'll see each subject code in that data, and we'll see that in a few minutes. We can snapshot our inputs if you want, so this is a reproducibility thing to make sure we can always come back to this assessment and look at the exact input data that was used. This is very handy if you want to be able to come back to a project after a year or some amount of time and really verify how you got the results you got, because you can see the exact input used. I'm not going to do that in this case because for a demo it's not really something we need, and any additional options we can specify here. Number of iterations, I'm going to leave it 1 for this case because it does obviously take longer the more iterations you get your assessment to do, and for the purposes of the demo it's good to make sure that it runs pretty quickly. So expert models is, as I've said, it's a cloud-based system, so when you run the jobs they're running on the cloud. This allows them to be quite large, so if we do really run a very high number of iterations, the system will be able to handle that. And we have a lot of models which use a huge amount of data, and they you get assessments which can run in less than a minute. They run very, very quickly. Obviously it depends somewhat on the complexity of the assessment, but there's others which will take hours, or even could be approaching a day to run if there's a huge amount of data and you need to do a lot of iterations. They really can take quite a while. So expert models will handle this because there's just the seemingly infinite resources of the cloud on which they can run. So we can see I've got a notification up here that my chemical exposure assessment complete. So the system's letting me know that my assessment's done, and it's just here in my file system beside all my data, which means it's in the same place as all my project work that I have in this folder. Okay, so I'm going to open that. As soon as I do, there's some different sections to our output. So you can see we've got some graphs here which are broken down by the stratifier that I created in my assessment wizard. So I asked it to be broken down between people who are 0 to 18, people who are 18 to 65, and then finally 65 to 120. So all of the normal calculations are done 3 times. This is a report, so it's a fairly shallow level of detail. It's something you can just look at very, very quickly and see your exposure values. The more detail that people tend to want is in the data tables, which I'll go into in 1 second. But before that, there's this inputs tab, which is very important. Again, this is to do with reproducibility. So we can see the tables that I used for the assessment. So for example, at any time I can go back to my foods table, which is in the system, and I can see the exact table that was used. Now someone could have edited this table since, and if we want to make sure that doesn't happen, if we want a full snapshot, that's when we would have checked that box I showed a few minutes ago, the snapshot inputs box, to make sure we have another copy which can never be changed by anyone else. So after we look at that, the most important thing then are to output tables. So this is very much similar to what Cian was describing a few minutes ago. There's a summary statistics table, which is always calculated, and then I checked a box for the subject data table, which is an additional calculation. So I'm going to draw a couple of graphs based on these, just to visualize what's there, because obviously big data tables, it can be hard to just wrap your head around what's in them straight away. But if we just take a quick look at them, so exposure type, we've only got 1 chemical in this assessment. We might have had multiple ones. You could run an assessment with multiple chemicals. In this assessment, we just have 1. So exposure type in this case is always going to be aspartame. Our group code, we've got 4 of those, and our group name as well. So I happen to make them the same in my data, but you can see which group we're talking about here. The model calculates various statistics, so you can see them here, things like count, but also the min, the max, and percentiles, and the mean. And then we've got our stratifier. So this is because I chose to stratify the output. So you can see some of the entries are for ages 0 to 18, ages 18 to 65, and then finally 65 up. We've got 2 calculation types, which is absolute and per unit body weight. So we'll see those when I graph in a second. But there's 2 different calculation types, and then we've got the output, the consumer exposure, so people who consumed the foods that I chose to run in my assessment, so the 56 foods across the 4 groups I put them in, and the total population exposure, so that's just averaged over everyone in the total population. And since I didn't filter my subject table, that's the total population from the subject table in this case. If we open the subject data table, this is a lot bigger, and you can see this has the subject code field. So this is data you can often do a little bit more with, because it's before you calculate your summary statistics. So you've really got the data for each subject in this. So you can see our first entry here. is for a specific group for this subject. You can see their statistical weighting and their body weight, and then you can see the 2 exposure values here, and you can step through. Whereas before we were looking at summary statistics on this, you can look at it for each subject, and we're calculating the foods value here as well, so their exposure from all of the foods as well as their exposure from just the 4 individual groups. And you will note as well, you might be noticing that there's not full entries, so not all of the subjects will have consumed all of the food groups, so there could well be missing entries, and that's a perfectly valid thing to have in here. So if we jump back to the summary statistics for a minute, I might just do some form of simple graph just to help visualize this. Like I said, it's a lot of data to kind of get your head around, but before we do that, we will need to filter it a bit. Specifically because we've got a lot of different statistics here and we can't really graph them, it wouldn't make any sense. So let's just look at the means for a second, just something simple. In our groups, let's look at, so we've got, let's look at our 4 groups, but not look at the total. So we won't look at the exposure to all 4 for this graph. We'll just look at the 4, and that'll allow us to see a kind of a contribution of those 4. And let's just look at one calculation type. So we are just going to look at per unit body weight instead of absolute, and then we're going to finish up here just looking at this graph. Okay, so that will allow us to look at this. So okay, so I apply those filters, that gets me down to some data, and then I can graph them, and we will look at our group. We will group by group name, and then look at our exposure. And we can very quickly see here. So here's our 4 groups, and here is the contribution to the total from all 4 of them broken out. So it's a quick way to kind of visualize the data, and you can do this very easily. There's all the graph types that I showed earlier as well. So we could look at a box plot, or we could look at a histogram, and you could also do this with the subject data table, which might allow you to do slightly different things as well, because you've still got the data at a subject level. So that was a very quick tour, and I'm conscious that we're just running out of time. That was a very quick tour of expert models. So there's huge complexity to the models, there's huge complexity to the software, and I appreciate that was an intentionally shallow enough demo just based on the amount of time that we have. But I hope that it covered enough. Fortunately, if anyone has any questions, you can contact me or Cian or both afterwards. I'm just going to open up our questions panel to see if we have any. Okay, so if anyone does have any questions, there's I suppose 2 things you can do. You can either type them in the chat window now, and we'll address them. That is public, so everyone will see them, but we'll address them here. Or you can email. So I'm going to finish. Oh, I went back to the start of Cian's presentation. Apologies. And I'll just jump to the end there where we have the email addresses, because you can get in contact. And I'll actually, I will maybe paste these email addresses into the chat window there, so you can easily copy them afterwards. But these are the email addresses, so if you do have any questions and you want to follow up, by all means get in touch. Otherwise, if you want to write any questions in the chat window, go ahead and do that now. Okay, I'm going to assume there's a raft of questions coming in over email, and I'll expect them and set some time aside to reply to them all. So thanks everyone for tuning in. We got a really good number, and I hope everyone enjoyed it. Do send on questions. So I'll leave the email addresses up there on screen. You can take them down, and do send on any questions you have about either Cian's presentation or the demo itself, or further things about expert models. It is a platform for hosting models generally, so we've just shown one of them today, and just gonna
and just say thank you all for your attention. If you have any questions on anything I've discussed, any feedback, we'd love to hear it. Like Mark was saying, we just demonstrated one of the models, but it's our platform now for any kind of predictive model that we generate. So If you have any thoughts or queries, please do let us know. All of us, no, these are just thank yous. Good. So thank you all for your attention. Feel free to follow up by email and have a good rest of your day.
Thanks, everyone.